Okay, welcome everybody to our keynote speaker. It's really a pleasure to welcome David Baker back to the LNB. I know he's connected online, but we'll just see his screen in a minute. David is the director of the Institute of Protein Design. He's a Howard-Hughes medical investigator, professor of not only a professor of biochemistry, but an adjunct professor of genomic sciences, bioengineering, chemical engineering, computing sciences, physics, all at the University of Washington. Not many people know that David, though he's known for computing some proteins, started his PhD on membrane trafficking with Randy Shackman, working on the sex system. He then moved on to David Agard to do his postdoc on fundamental in vitro protein folding, found how a pro-region of the alpha-lytic protease kind of overcame a kinetic barrier for the folding. When he founded his own lab in Washington, he had two ways of both experimental in vitro protein folding and also computational approaches. One of his early papers, and that was a seminal paper on contact order in protein folding, where you could actually see how topology affected the rate of protein folding. But of course, he's well known then for his ab initio protein structure prediction with Rosetta, and also distributed computing with Rosetta at home, and also Foldit. He's published over 550 papers. He's co-founded 17 companies. He's been granted over 100 patents, and 68 of his mentees have actually gone on to form independent positions. He's won numerous prizes, including the Breakthrough Prize in Life Sciences. He's a member of the National Academy of Sciences, the American Academy of Arts and Sciences. So it's a great pleasure to welcome David to talk to us again here at the LNB.
All right, well, thank you very much for the invitation, and I'm sorry I couldn't be there in person. I wanted to tell you about de novo protein design, which of course is the problem of designing brand new proteins that don't exist in nature. And in fact, none of the proteins that I described today have any sequence homology to naturally occurring proteins, so they're all completely made up. And I'm going to describe two approaches. For many years, we and our colleagues like Riju have been developing the Rosetta sort of physically based approach to protein structure prediction and design. And this is really based on the idea that proteins fold to their lowest energy states. And so in Rosetta, there are models for calculating the energies of hydrogen bonds, van der Waals interactions, and so forth, and methods for sampling through sequence and structure space. So to design a new protein in Rosetta, you come up with the structure you want to make, and then you find a sequence whose lowest energy state is that structure. You're all aware of the huge advances in deep learning, really spearheaded by John Jumper. So now that we have ways of mapping from sequence to structure, we can cast the protein design problem in a different way, rather than in energetic terms, designing a sequence whose lowest energy state is the target structure. We can instead cast it as design a sequence which is fairly strongly mapped onto the desired structure with the desired function. So the first part of my talk, I'll describe sort of where we are with sort of the physically based approach to protein design, and then the latter part, I'll talk about deep learning approaches. Let's see. All right, so it's an exciting time for protein design. We have our first approved medicine that's actually going into people. And it's actually interesting, the story here. So a number of years ago, we worked out methods for designing self-assembling icosahedral nanoparticles. And it was kind of cool, but we didn't really have a clear application in mind. And just a few years later, my colleague Neil King has found that if you place parts of a virus, for example, in this case, the receptor binding domain of the coronavirus on these particles, you elicit really, really potent responses. They're a bit more potent than the mRNA vaccines. And the really exciting thing is that these don't require, ultra low temperatures, are stable for a long time. So for distribution in the developing world, they have big advantages. So anyway, so de novo protein design was kind of like, it was kind of a lunatic fringe area for many years, but now it's actually turning out to be useful. We also have a number of other design proteins in clinical trials now. So hopefully I'm optimistic that we'll see a transition from protein therapeutics being discovered by random selection or by fishing things out of the body to really intentionally design proteins. So a first example of such a thing is our rather than coronavirus vaccines, therapeutics. So Longjing Cao and Brian Coventry worked out very general ways of designing proteins to bind to arbitrarily selected sites on any target of interest. And they applied this method to the coronavirus receptor binding domain. And so here you see the spike trimer, and here are three of this is a cryo-EM map showing the three of these domains bound simultaneously. And a comparison between the cryo-EM map and the computational design model shows that the design is very, very accurate. So we can so this protein is very potent against the wild type virus. To make it more potent against sort of escape variants, we took advantage of avidity and basically designed this tripodal structure with three receptor binding domain, three of these mini binders. And here you see the cryo-EM structure in complex with the spike trimer, and you can see all three of these domains are engaged simultaneously. And this shows really potent neutralization and protection in animals against all escape variants tested. So this is headed towards clinical trials, hopefully pretty early next year. So I should say that now that the methods for designing these binders have really gotten almost routine, we're making small proteins that bind very tightly to the major cell surface receptor subunits, and then we can array them in different ways as sort of we can bring same copies of one receptor binding domain or different copies of different or multiple copies of different receptor binding domains together. And we're seeing very interesting effects on cell signaling. So I think this will be a very powerful route to new types of agonists and bias agonists. So we've gotten pretty good at designing binders to relatively static rigid structures. What about the flexible peptides? Well, Danny Sato developed an approach. This is to bind flexible peptides, assuming that they populate some fraction of the time a beta strand-like conformation. So what he did was to design proteins which basically have an empty slot for a beta strand, as you can see here. So the side chains that stick up here make very strong packing interactions with the rest of the protein. So they kind of complete the beta sheet. And he's made quite a number of these, and they bind quite tightly. And with Thomas Knowles at Cambridge, we have Thomas's lab has these very exciting results. Taking one of, he's done this with multiple of Danny's binders, but this just shows data for one of them. So his group has developed methods for monitoring a beta aggregation, and upon addition of stoichiometric amounts of Danny's design, this is completely suppressed. Now, how interesting and relevant this is therapeutically depends on when you read the news. But as of this morning, the idea of going after amyloid precursors directly is looking pretty promising. So we're quite excited about this. Danny has made binders to all of the major amyloid-forming proteins, and there certainly are diagnostic, if not therapeutic, applications. So just to show how these kind of techniques can be used outside of the medical realm, this is still on the theme of binding, but a rather different problem. Nate Ennis has been really fascinated by the early stages of photosynthesis, which involve absorption of light by the special pair of two chlorophyll molecules. Now, this is a very complicated process which goes on inside a really big, complicated protein. So what Nate did was to design a dimeric protein shown here that holds these two chlorophyll molecules in nearly identical geometry that they have in photosystem. And so he has crystal structures now, actually have multiple designs where the chlorophyll molecules actually have slightly different geometric relationships with them. And now it's really fascinating because he and his colleagues can do photophysics to sort of determine exactly, how the level of exciton coupling and so forth depends on placement. Now, working with Shunji Wang, Nate assembled these into these octahedral nanocages, which are shown here. So in Shunji's design, we have a design trimer here, and then we have Nate's C2 structure with 2 chlorophyll molecules. So in these resulting cubical structures, which are really quite beautiful, you can see them here, there are 2 chlorophyll molecules on each edge, and we're really excited about building synthetic light harvesting systems extending on these ideas. So I told you that almost everything we're doing is with proteins that aren't related at all to what exists in nature, but in some cases, we actually do incorporate naturally occurring proteins. So in this case, the yellow protein is an antibody, and Robbie Devine designed these different green proteins. This is a pentameric protein that binds antibodies and wraps them into icosahedral nanocages. Here's a green tetramer that wraps antibodies into octahedral structures. And the beauty of this is you can substitute any antibody in and generate a nanoparticle that contains it by simply adding the correct protein here. And we found that the agonistic activities of many antibodies are enhanced by incorporation into these nanoparticles. This is an example here with the death receptor antibody, which has relatively little effect on cells in isolation, but potently kills tumor cells when it's wrapped into a nanoparticle. Now, an attractive application of these is delivery, because we have the antibody combining sites sticking out. And so in principle, you could package whatever you wanted inside the nanoparticles and then deliver it to a location in the body just by incorporating the right antibody. The problem, though, is that there are these empty channels here. And Erin Yang, a graduate student in the lab, took on this challenge. So she designed these trimeric plug-like structures that fit within these open pores in these octahedral nanoparticles. And this is a cryo-EM structure solved by Andrew Borst, where you can see down here's a 3-fold axis, Erin's plug, the 4-fold axis, the designed protein, and the C2 axis where the antibody sits. In this case, it's just an FC. And the neat thing is that Erin has designed this plug to be pH dependent, and she has different versions which are come off at different pHs. They're loaded up with buried histidine residues. That's the basic principle. And so this is well within the range of endosomal pH. So the idea is that you can target these to any cell you want by making them out of the right antibody. And then when they get endocytosed, the plug will come off, as shown here, and then the contents are released. And Isa Yosef, a graduate student in the lab who just graduated, has some very exciting preliminary data on guide RNA delivery using this mechanism. So we've also been extending these methods to unbounded materials, and in particular, in this case, crystals. So Zhe Li and Shunji Wang and Una Natterman have developed very general methods for designing protein crystals. Of course, there are hundreds of thousands of crystals that have crystallogers have grown, but now we can actually intentionally design different crystal lattice forms. And here you see 3 different examples. We can, of course, compare the designed crystals to the crystal structures that we solve of these crystals. And in general, they're quite close. So this really opens up new opportunities for solving structures by host-guest methods. We can also package various things inside, like nanoparticles inside these cages, and have them regularly arrayed. So I think there's a lot of exciting materials design applications here. So everything I've talked about so far is a transmembrane protein, but Anastasia Vorobyeva, when she was a postdoc in the lab, really pioneered the application of these methods to the design of beta barrels, and in particular to transmembrane beta barrels. And Samuel and Sagardip have been continuing, postdocs in the lab, and continue to work with Anastasia and her group on now functionalizing this. So here I first want to show you a crystal structure of a 10-stranded transmembrane beta barrel. This is purified from detergent. Here's the design model. You can see they're very similar to each other. And this is a design that has a very nice NMR structure. This is one that Anastasia designed. And the really exciting thing about this is it has very nice single channel conductance activity. So we're very excited now about being able to now functionalize the interior of this pore for all kinds of different applications. Sequencing methods have been based on naturally occurring pores, but now that we can design them to spec, I think there's a huge number of opportunities. Now switching gears a little bit, proteins, I think when the first crystallographers like Max Prout solved the structures of proteins, it was kind of disconcerting how irregular and asymmetric and complicated they were. And that makes it really hard for, non-biologists to really, look at protein structures or to do things with them. But Tim Huddy, a grad student in the lab, thought that this really didn't have to be the case. So he set out to design a new generation of protein building blocks that really makes a protein design like Lego. And here are some of the things he's designed. So the basic idea is to build structures out of perfectly straight helices, as illustrated here, such that there's, in this class of building blocks, they're really perfectly straight and there's no twist at all. And here's a crystal structure. He can also design, by incorporating rotations in the helices, very discrete sorts of bends. And here are structures of a triangle and a square-like structure that he's made. These are all cryo-EM structures using this technique. As I'll show you in a moment, you can just include more repeat units to expand or shrink these structures. And here, then you can also introduce smaller, he and Jinwei Zhu have worked out how to introduce smaller changes and rise to this continuous curvature. So Jinwei's designed these really kind of amazing rings of different sizes. These are just negative stain EMs, but we have higher resolution cryo-EM structures. These form incredibly homogeneously. Tim and Yang have figured out how to place these units at defined angles relative to each other, which makes it really straightforward to build up different types of nanocages, as shown here. And they really look very different from naturally occurring protein structures. So here's a straight tetramer that now makes a 90-degree angle here. And so we get these basically cubic assemblies. And the beauty of these structures is they're kind of extendable nanomaterials, because you can just insert additional units here to go from like this small cube to a medium-sized cube to a big cube. Also using this technology, it's very analogous to like building things out of, lumber. You can add in reinforcements like these struts showing here. This is a sort of a 1D essentially assembly of these helices, and then Tim's built in these struts that make interactions on both sides. So this is overall C2 symmetry, and Tim produces these very beautiful train track structures, which again, it's very, very tunable in terms of the spacing of the struts and the length of the struts. So we can make a wide variety of structures, not by doing any complicated protein design, but almost by inspection, changing the number of helices. Now, with the rings, similarly, Jinwei and Tim have developed methods for placing rings within rings, all using these same basic concepts. And these have really kind of remarkable structures. So here are EM projections of this 2-ring structure, and here's another one. So using this approach, we're very optimistic about being able to sort of very quickly design a wide variety of protein nanostructures. Again, that will look much more like things in the macroscopic world than kind of the unusual twisty protein structures. So nature didn't really worry about the ease of building, but I think for people this could make a big difference. So everything I've talked about so far is are static proteins, but what about dynamic proteins? And in particular, there's been a lot of interest in metamorphic proteins, which could adopt 2 different states. So Florian Praetorius and Phil Leong took on this problem. And so the basic idea is to design a protein that's 2 states, again, similar to what I just showed you, sort of made out of helices represented by these beer cans. And by twisting around one helix, this protein can adopt this other state. And the basic concept for driving this conformational equilibrium is to bind a a ligand, in this case, another helix that pulls the equilibrium. So this has really worked beautifully. So this is a, we call state, the closed state that doesn't bind the peptide is state X. State Y is the peptide bound form. And we now have crystal structures of these designs in both forms. So they populate one structure in the absence. Here's a crystal structure in the bound form. So here's the ligand peptide bound, and here's the open state that's got the peptide bound to it. And we just got a crystal structure in the closed state, and it's exactly as designed. So this system really does have two different states. We have some systems that that at equilibrium just sample both structures and then can be driven into one of the states by adding the peptide. So one way to follow the conformational change in real time is with Deer spectroscopy. So here we have in the apo structure, these two introduced spin labels are far apart. When the peptide binds, there's this conformational change happens, and they are driven closer together. And here you can see in in these Deer spectra that the distances between the probes become much less when the peptide is added. We can also follow this by FRET and other techniques. So we're now working to be able to cyclically drive this kind of conformational change as the basis for protein walkers. But today, what I want to show you is a different application of these, and that is to the general problem of allosteric control of protein structure. So, Arvind Pillai and Abbas Idris have basically taken these sort of two-state proteins and built oligomers out of them where one state can form one oligomer and the other state, when the peptide bind, can form another. So I'll give you a couple examples. This is a trimeric state that Arvind designed, a trimer where where in the state where the peptide isn't present, it forms this trimer. When the peptide is added, then the units change, and instead a dimer is formed. And you can see that here on the sec column, when a peptide is added, there's this pronounced shift to lower molecular weight. And I'm not showing it, but we now have very nice EM data showing this shift between conformational states. And here's another case where it's going in the opposite direction, where we have in the unbound state, it's a C2 structure. And when the peptide is added, it goes into this trimeric structure. Arvind has also designed systems where the peptide drives a transition from a monomer to a oligomer structure. And what's really neat about these, they're sort of classically allosteric in the sense that the peptide is not binding at the interface. The peptide is binding distance from the interface, but it's causing a conformational change that then allows binding or assembly. Okay, so that's everything I've described so far was done using Rosetta and classic energy-based design. In this case, here we're trying to design proteins that have roughly equal, for these switches, roughly equal energies. So really inspired by John's work with AlphaFold, we developed a network called Rosetta Fold. And some of the interesting things about this, it's got a slightly different architecture than AlphaFold. It's got this third track. It scales really well to very large systems. We're about to release the latest version, which is which is comparable to the published version of AlphaFold. I've no doubt that John and his colleagues have better versions now on monomers and multimers. So it has the advantages that it scales really well. And also, Frank DeMaio has generalized this to nucleic acids. So we've now made this available both to RNA molecules, and Riju, will be really exciting to talk about many of your new examples, as well as to protein nucleic acid complexes. So I encourage people to try it out and give us feedback. We're really excited to see what sort of problems it can help solve. We've also developed methods, I apologize for how busy this slide is, for doing the inverse problem, rather than going from sequence to structure, going from structure to sequence. So here we take a structure, and it's basically solving the same problem that Rosetta sequence design solves, where you find the sequence predicted to fold into that structure. And it's been a little disconcerting for those of us who have been working on developing Rosetta for a long time. So this is the sort of the longstanding benchmark for protein sequence design, which is given native structures, how will you recover the the native sequence, native backbone. And so this problem gets harder as you move away from the core of the protein. So in deeply buried positions, you can predict the sequence pretty well. As you go further away, it gets harder and harder. And but this protein MPN network really does better than Rosetta all across the board. And we've also gone back to designs that we've made using different methods, for including Rosetta, that didn't work, and redesigned them with protein MPNN. And now they work, which is, so we've made protein MPNN also freely available. So just a couple cool things that we can do with all these methods. One is to just explore, what's possible, explore sort of protein space in an unbiased way. And we call this protein hallucination because it's a little bit analogous to the way that, given a network that is trained to classify images of cats as cats, you can ask it, well, what does an ideal cat look like? And it will spit out images. You're probably familiar with Deep Dream and Inception. Sometimes they look like cats, sometimes they look much more exotic. So we wanted to try, basic idea was to try something similar with proteins. So in this case, this is work from Lucas Miles and Basil Wiki. They basically chose, and here they choose a length of a protein, L, and here they were interested in oligomers, and oligomer symmetry, or number of copies, N. And then they choose completely random sequences, predict the structure, and then evaluate, giving it, in this case is with AlphaFold, giving, say, 3 copies, evaluate the extent to which this forms a cyclic trimer. And of course, at the beginning, it won't really form a cyclic trimer, but then they can optimize the sequence till the point, just by doing optimization in sequence space, to the point that the sequence is strongly predicted to form a trimer. And this is a case where the initial sequences we got out, it's well known that these sort of expectation maximization methods can lead to adversarial sequences, didn't work. But when we use protein MPNN, they expressed beautifully, and we were actually able to solve crystal structures of 7 of these. Again, we put nothing in except for a random sequence of it. We chose the length and the oligomerization state. We got all these cool, quite non-trivial structures out, as you can see here. These are trimers and tetramers, and these are just comparisons of the crystal structures to the hallucinated models. And Alexis Courbet who had been working on making protein machines, was interested in seeing whether he could make sort of larger machine components using this approach. And so he went up to 33-fold symmetry. So basically generating a random sequence, tandemly duplicating it 33 times, or basically copying it 33 times, and then predicting the structure, and then going through the same optimization. And you can see here cryo-EM structures of these designs which he made, which are really quite remarkable. They're perfectly circular, and they have very high symmetry and quite large diameters. So I think this sort of illustrates how deep learning methods can be kind of used in an unbiased way to explore the protein structure universe. We can also use these methods for de novo enzyme design. So here is a luciferase reaction on a synthetic substrate that doesn't exist in nature. And Andy Ye and Chris Norn were interested in designing enzymes which operated on this substrate, and so they Chris designed use these deep learning methods to design a series of scaffolds that are shape complementary to the transition state for the reaction. And then Andy used built-in active sites, in this case using Rosetta still, that stabilize this transition state. And what came out of this were some very active luciferases. So this is just a tube with cells that are expressing the luciferase versus not expressing the luciferase. This is a stepping stone along the way. So in the past, to get really high activity, collaborators like Don Hilvert and Danny Tofik had to do really extensive directed evolution. Here, with just 2 substitutions from the original design, Danny was able to obtain enzymes with kcat over km of 10 to the 6 per mole per second, which is really quite respectable, and on the same scale as naturally occurring luciferases. Now, the activity is on the same level as the naturally occurring luciferases, but the naturally occurring ones have these really big open pockets, and So they don't really have much substrate selectivity. This is over this battery of different substrates here. But Andy's design, as I showed you, has this really closely packed design pocket. And so it is very selective for the naturally occurring substrate. And so Danny is now making what we hope will be similarly selective designs for these other substrates. And we hope that will allow multiplexing of luciferase imaging in the same way that one can now do with fluorescent proteins, because essentially you can have different colors in the same cell. Okay, and so my final topic is sort of general function design, protein function design using deep learning. So here, and the basic idea here is to scaffold an active site or a functional site. So given a description of, say, a metal binding site or an enzyme active site, or a site that would elicit neutralizing antibodies, or a protein-protein interaction site, and there's a general problem in protein design, which how do you design a protein which has that site? So we've developed two approaches to doing this. One is constrained hallucination, which is a very similar optimization in sequence space, except in this case, not only just optimizing the sequence such that it will fold and assemble into, for example, the desired assembly. Instead, we also favor the folding to a structure that contains the desired site. And that's illustrated here in the case of immunogen design. This is the respiratory syncytial virus F protein. And here are two epitopes that Bruno Carrera's lab has identified that are very important that are targets of neutralizing antibodies. But the protein's really big, so we want to design small proteins that only contain these epitopes. And so, so basically, these are examples of hallucinated structures which contain these epitopes, which almost perfectly recapitulate these epitopes. We've made these proteins in the lab, and they bind the antibodies. And we've actually now been able to extend this to build proteins that contain multiple epitopes, all in one small protein. And Bruno's lab is currently investigating their effectiveness as vaccines. The second approach is kind of an information recovery or approach. And the basic idea is you're all familiar with how when you type a few words into a word processor, it will attempt to complete your sentence after it has enough information to guess what the next words will be. And that's because it has a language model built into it. And so we trained Rosetta Fold by sort of randomly masking out different parts of the sequence and structure of proteins in the training set. And it got very good at building in the missing information. And so now we have, and this allows a very rapid way to solve this protein functional site scaffolding problem, because we can just provide the functional site like this, and then build out the just auto-complete the rest of the protein. And again, we don't really specify anything about it. The Rosetta Fold just fills in the sequence and structure, and we get quite a variety of different solutions. This just shows the application of this method to metal binding sites, where we, again, experimental characterization shows they're quite functional. But since we've extended this to a wide variety of things, making new types of cytokines, other types of binding molecules, it's a very powerful approach and very fast, because the auto-completion just takes one pass through Rosetta Fold. So thanks for your attention. I've had really fantastic colleagues on all of this work. So Longjin and Brian developed the general protein interaction design binder method. Keija developed those trimeric coronavirus binders, and the whole coronavirus binder project has been a huge collaboration with Mike Diamond's group, Andrew Hunt in Mike Jewett's group, and it's been a fantastic thing. Danny Sato designed the amyloid binding proteins, and again, it's been a great collaboration with Thomas Knowles. Nate Ennist and Shunji Wang designed the light harvesting, the chlorophyll binding proteins in those cage-like assemblies. Aaron and Robbie and Andrew did the work I showed you on the antibody cages for delivery. Zhe and Shunji and Una designed the protein crystals. Tim and Yang designed those extensible straight protein materials. Flo, Phil, Arvind, and Abbas designed the two-state protein switches and the allosteric assemblies that it built from. I didn't have time today to talk about molecular machines. The protein hallucination, The cyclic oligomer hallucination work I showed you was done by Alexei with 2 rotation students, Elias and Sam, and Basil and Lucas. Rob Ragot solved the crystal structures. The protein MPNN is the work of Eustace Dupras. Frank and Mink Young developed Rosetta fold, and again, we're about to release a new version, but I encourage you to try out the protein nucleic acid version that's available now. Andy and Chris designed the new luciferase, and Ju and Sydney, Doug, and Sergey developed this constrained hallucination approach to functional site scaffolding, and David and Joe developed the in-painting approach. So thanks for your attention. I'd be happy to take any questions.
I'm sure we're going to have a lot of questions, and I see there's one right at the back first. Yep.
That was a fantastic talk. I was wondering whether you could comment on the ease with which some of these design proteins can be produced. And if you're able to design proteins that can be easily produced, could you take those learnings to things that people might want to produce that could be altered a little bit to be made
Yeah,
easier?
that's a really good question. So not all the designs work. And so my talk was very misleading in the sense that I showed you, data on these designs which functioned as designs. Many designs, when we express them in E. coli, the bacteria don't like to make the proteins for whatever reason. But I should say, first of all, the access rates are really increasing very, very dramatically. And the proteins that do express tend to be expressed at very high levels. They're the major protein in E. coli. And often we can purify them simply by heating the bacteria, and all that's left is the design protein. So for example, for the coronavirus therapeutic we're developing, the little protein that binds to the coronavirus, you just take E. coli lysate, take E. coli that are expressing the protein, boil the E. coli, spin, basically you have protein that's pure protein than the supernate. And these methods, some of these things can be transferred to proteins that are naturally occurring proteins. We've had good success in increasing the expression levels of a number of proteins. So if you're interested, definitely send me an email. We're still working out the best way to do this, but I think it's a very exciting area.
Okay, we've got one online.
We have one question online. The question is, how are the novel proteins expressed and purified?
Yeah, well, most of the proteins are expressed in E. coli, and we usually just put a his tag on them. And so we purify them in one step on a nickel column. Sometimes we run an SEC column after that. For some of the cases, we display them on the surface of yeast or express them in mammalian cells, depending on what the goal of the experiment is. But E. coli is really the workhorse. You can also express them in E. coli in vitro, and they express very well.
Okay.
Hi, David. So I'm curious what you think about the pros and cons of building sort of nanoscale modules with the sort of principles of macroscopic assembly, and what sort of functions you think you'll be able to access on a nanoscale, and whether that's a sensible principle to build biology with, or what you see the opportunities and limitations of sort of pursuing that strategy.
Well, I think it really depends on what the goal is. For regular architectures, like if you want to array, anything of interest out in 2 or 3 dimensions, I think these really straight building blocks are very useful. And it's also just very nice to be able to build things almost by inspection. Obviously, if you want to design catalysts, that's not going to be a good way to do it, because it really depends on the details. One of the things we're working hard on, I think one of the problems with building things out of proteins is essentially just for real world applications, there's a limit to how much protein you can make. So we're working on sort of integrating these protein design methods with inorganic materials, for example, silicon nitride with drilled holes in it, where you could place some of these arrays. So I think the answer to your question is really going to depend on the problem, but we're already seeing that with these sort of really kind of Lego block-like proteins are really making designing molecular nanostructures much more straightforward.
Hi,
Yeah, Tony,
it's
Mike.
fascinating research. I just want to ask, what is the application of the triangles, squares, and rectangles that you have designed that are so pretty? Or this was just a folding exercise? And the second
Yeah.
question is, yeah, I have two, sorry. The second question is, how do you trick the immune system when you design proteins? Because they are immunogenic, yeah, so, and then how do you, can you predict actually the immunogenicity of protein which you design?
Yeah, well, those are both really good questions. As far as the applications of those squares and triangles and train tracks, we're trying to answer that question ourselves. So if anyone has any, so yeah, I mean, our hope, so far it's really just demonstration. I mean, sort of the hope is that if we could really get a protein toolkit that enabled building any type of structure really as easy as it would be with, you know, any kind of sort of kids building set, then there will be applications where, you know, one of the things we're thinking about is arraying lanthanides in regular arrays. There'll be probably more materials applications than biological applications. So on the immunogenicity side, that's really kind of the $6 million or $6 billion question right now. We're going to learn a lot about that because we have several proteins, designed proteins in clinical trials now. For example, a cancer therapeutic based on IL-2. What I can tell you is that we've done pretty extensive preclinical experiments with the coronavirus binder and various other proteins we've put into animals, and we've seen pretty minimal immune response. And that's for these the small binding, sort of what we call mini binders. And that's, I think, because they're very stable, very soluble, and it's difficult for dendritic cells, just don't take them up very readily and break them down. But of course, humans are very different. So we'll be, you know, I think the jury's out. We also have been doing some work on predicting immunogenicity using deep learning methods. We just put a preprint out on that, but it's still not clear whether we're going to have to worry about engineering out T cell epitopes or whether just the biophysical properties of these proteins will be sufficient to make strong immune responses rare.
Hi, great talk. So on protein MPNN, I was just wondering if it generalizes well to flexible proteins such as antibodies, let's say, things like that.
Well, it requires you know, what you input is a single structure, so it will design a sequence which is stable in just that structure. It's, you know, it's a purely backbone to sequence method. So so if you want to sequence, so one of the things that the protein MPNN is good at is you can give it multiple states. So for example, the way we're designing those switch-like proteins is to provide both states and do multi-state design. So to find sequences that have are strongly predicted to be in one of the two states. But if you have a disordered protein, then there's not really anything for the protein MPNN to work with. But you can certainly stabilize particular conformations of proteins.
Thanks.
Yeah, we just got one online. If we just wait for the microphone to come down.
So there are two questions online. The first one is, how do you think the physics-based approaches will play out with deep learning in the coming years? Are you thinking on deep learning models that will learn protein physics from your energy functions?
Yeah, that's a good question. I think the most obvious, the deep learning methods, as has been mentioned, really requires a lot of training data and, you know, a lot of evaluation and testing data. So when you get to situations where that doesn't exist, for example, proteins with unnatural amino acids, or in our case, we've been doing a lot of work I didn't describe on taking the design principles, the sort of the Rosetta-based design principles, and applying them to macrocycles, and we can build now essentially small molecules out of different types of backbones and side chains. And in those cases, there's really not the training data. They're not just made out of the 20 amino acids. So you can't use things like AlphaFold and Rosetta fold. So I think basically regimes where there isn't much data are places where the physically based models are still very useful. And of course, you know, we're trying to put more and more of the physics into Rosetta fold. You know, and so one can try and incorporate the whatever physical information you have. And again, that'll probably be particularly useful in low data circumstances. But as far as, you know, you can't train a neural network on energy. It's not an observable. And so so there's going to be so I think getting new physical insights from deep learning methods about the physics is going to be, it's not totally straightforward.
Thank you. And then the last question online is there any shared characteristics among the proteins, design proteins, that do not express well?
Yeah, that's a good question. I mean, we've looked quite a bit at our design proteins which fail to express and don't express well. And, by and large, it's, one of the things that's actually a reasonable predictor of function is you just give the sequences to Rosetta fold or AlphaFold and ask how well they're predicted. This was for the original Rosetta designs, but we certainly get lots of designs that are, say, strongly predicted by AlphaFold or Rosetta fold to fold to their target structure that don't have the desired function and don't express very well. So it could be that there's something in the sequence that that the bacteria don't like. It could be that we still have that, despite all of our in silico metrics, there's still something fundamentally wrong with the design. So it's hard to answer that question. I mean, there are many sources of failure if you just think about the biosynthetic process, transcription, translation, stability. So we don't really know what the cause of most of the failures is.
Okay, so maybe we'll just have, we've got two questions now. Can we have a microphone just here?
Hi, David. You mentioned going beyond the confines of the 20 amino acids that we all know well. How do you propose, in the absence of sort of training data and things like AlphaFold, you overcome sort of the potential experimental hurdle of how do you validate these things when bacteria, for instance, won't know what to do when you're faced with this novel amino acid?
Yeah, that's a good question. Well, the approach we've used is just chemical synthesis. So we've really focused on smaller systems, basically with between 4 monomer units to about 13 or 14 monomer units. And these monomer units can really be anything. They can be alpha amino acids, beta amino acids. They don't have to be amino acids at all. And we've gotten quite good at, and this is using sort of the traditional Rosetta approach, where you're calculating energies, enumerating ensembles, and checking that your design sequence has lower energy than your design structure has lower energy for that, chemical structure than anything else does. And we just published a paper in Cell last month showing that we can very systematically design structure, and that these design structures are membrane permeable. So so we're back kind of in pre-deep learning. We're trying to extend our deep learning methods to these macrocycles with some success, but so far, just the Rosetta-based principles have worked out pretty well for these smaller structures. But yeah, we can't make them in cells because they have many different types of backbones and unnatural amino acids, side chains.
That's good. And I'll just let Paul ask the last question of the meeting.
Yeah, David, I mean, you've got a great luciferase, which is an impressive enzyme, but your structural energy wells are very deep and narrow, which is why they're so stable. Can you tune the design so that you can have broader wells, so that you can access more closely related structures and hence have a more dynamic structure for catalysis?
Yeah, I think that's one of the really important open questions in protein design, which is, yeah, so we've gone pretty far with designing these proteins in single deep wells, which is why they're so stable. But for enzymes, you need to be able to, bind the substrate, selectively stabilize the transition state, release the product. So I think it's not so hard to design proteins that, are more flexible. I think what's really hard is to design in exactly the right dynamics. So you want sort of dynamics along the reaction pathway, and that will help you do all those things. But you don't really want dynamics sort of orthogonal to it, because you'll lose binding free energy, or say transition state stabilization. So so if your protein is moving around too much, and it's very rarely in the configuration needed to stabilize the transition state, that will be a bad thing. So I think that is exactly the frontier here. How do you design in the dynamics needed for really high, really potent catalytic acceleration? And that luciferase example I showed you is really the easiest possible case, because that's not a very difficult reaction. But naturally occurring enzymes can stabilize, can increase reaction rates by over 10 to the 10th fold. And to do that, I think will really require mastering this problem.
Okay, thank you very much, David, for just a very, very enlightening talk. We're going to look forward to see where you go next